Welcome student. In the last lecture or last video, I have discussed I have discussed about the measurement setup of ion selective electrode. Now look how this ion selective electrode works. Okay. So see, there are commonly more than one type of ions present in the analyte or solution. Okay, so how this ion selective electrode manage to measure the concentration of certain ion in solution without being affected by other ions. Okay, so see, this problem is done by applying a selective membrane at the ion selective electrode, which only allows the desired ion to go in and out. At equilibrium, there is potential difference existing between two sides of the membrane, and it is governed by the concentration of the tested solution described by Nernst equation. Okay, so the Nernst equation is this: E equal to e to the power 0 plus 2.030 capital R into capital T divided by n small n into capital F log C. Okay, so where capital E is the potential, e to the power 0 is a constant characteristics of a particular ion selective electrode. R is the gas constant, this is 8.314 joule per Kelvin mole. T is the temperature, which is in Kelvin. N is the charge of ion, and F is Faraday constant, and this value is 96500 coulombs per mole. Okay. To make it relevant, the measured potential difference is proportional to the logarithmic of ion concentration. Okay. So thus the relationship between potential difference and ion concentration can be determined by measuring the potential of two solutions of already known ion concentration and a plot based on the measured potential and logarithm of the ion concentration. Okay, so based on this plot, the ion concentration of an unknown solution can be known by measuring the potential and corresponding it to the plot. Okay, so in this way this ion selective electrode works. Okay, so see, here I have summarized the working of ion selective electrode. So how it works? So see here, the cell consists of both an indicator and a reference electrode. Okay, see, the cell consists of both an indicator and reference electrode. Since the potential of the reference electrode is constant, the potential developed at the indicator electrode that contains information about the amount of analyte in a sample. During the measurement, there is little to no current flow. Okay, an electrochemical cell for making a potentiometric measurement with a membrane electrode, also known as ion selective electrode. So a specific ion electrode will only respond to the presence of one specific species. Okay, in reality, ion selective electrode can experience interference by responding to the presence of other ions. Okay, the selectivity of the ion selective electrode is determined by the composition of the membrane. Ideally, the membrane allows the uptake of only one specific ion into it. Okay, so there are three main components of making a measurement at an ion selective electrode, and these are an inner reference or standard solution. This is first. Second is an outer analyte or sample, and third is solution separated by a thin membrane. Okay. So the potential developed at the membrane is the result of either an ion exchange process or an ion transport process occurring at each interface between the membrane and solution. Okay. So at equilibrium, the membrane potential is mainly dependent on the concentration of the target ion outside the membrane and is described by the Nernst equation. Briefly, the measured voltage is proportional to the logarithm of the activity of ion in solution. The sensitivity of the electrode is expressed as the electrode slope in millivolts per decade of activity per concentration. Okay, so thus the electrode can be calibrated by measuring the voltage in solution containing, like example, 10 ppm and 100 ppm of the target ion, and the slope will be the slope of the calibration line drawn on a graph of millivolt versus log activity. Okay, so thus the slope simply equals the difference in the voltage, like log 100 minus log 10. Okay, and this is one. So unknown sample can then be determined by measuring the voltage and plotting the result on the calibration graph. The measured slope can be used as an indication of the proper functioning of an ion selective electrode. Okay. Now see, this figure represents electrochemical cell for making a potentiometric measurement with an ion selective electrode. Okay. So as indicated by their name, ion selective electrode possesses a high degree of selectivity. The selectivity of the ion selective electrode is determined by the composition of the membrane, okay, which I have already discussed. Ideally, the membrane allows the uptake of only one specific ion into it. The analyte ion may be a cation or an anion, okay, and the three main components of making a measurement at an ion selective electrode are inner reference standard solution, and second is outer analyte or sample, and third is solution separated by thin membrane, okay, which I have discussed in this slide. Okay, and this component okay, so and these components have shown in this figure. Okay, so the potential developed at the membrane is the result of either an ion exchange process or an ion transport process occurring at each interface between the membrane and solution. Okay. Now in the next lecture we will see the ion exchange process and ion transport with an ionophore. Okay. So today I stop here.